And welcome to the Evidence Reasons Academy. I'm Salvador Cordova, and I'm joined by my co-host Cindi Lincoln. Cindi, you can unmute yourself, and we're going to study molecular biology basics. And there's a little bit of an echo. I'm sorry. Your microphone is very sensitive. It picks up my sound. And unfortunately, you have to turn it down so low sometimes you can't hear me. But it sounds better now. So do you remember can you summarize what the central dogma of molecular biology is?
DNA splits or replicates into mRNA, which creates proteins.
Pretty close. So let us go to the National Human Genome Research Institute of the National Institutes of Health. They're pretty good as far as teaching materials. I mean, because the NIH is probably the biggest biology organization on the planet. If it's not, it's pretty close. So the central dogma says, the central dogma of molecular biology is a theory stating that genetic information flows only in one direction, from DNA to RNA to protein, or RNA directly to protein. So you summarize it. You were close. So DNA transcribes to RNA, and the proper term is it transcribes to RNA, and there's some steps in that. And RNA is then translated to a protein. So there are numerous ways that this is stated. So, but you had the basics, the correct order. DNA,
Sal,
yes.
the whole origin of life experiments where they make basically black tar in an attempt to make the building blocks of life. What you just read says it only goes in one direction, from DNA to protein. But weren't they trying to create proteins?
Without DNA, that was some of the origin of life experiments.
So proteins are worthless without a DNA. Is that
eventually, because you lose the recipe?
Well, and you can't go backwards. It just said what you just read said it goes one way.
It's problematic. And this is why it's good you're learning this, because you could see a flaw in one of R.N. Ra's emails to us. Some people said there are multiple peptides in the first biotic organism. Just like, well, that's kind of useless because there's no way you could store the recipe. See, the reason DNA is important, it's a good chemical to store the recipe, the blueprint. Because there's that song.
So proteins cannot replicate themselves.
What?
Proteins cannot reproduce themselves. They have to have DNA to do the duplication.
That's correct. Unless you have some other kind of machinery, and big machinery, it's very hard to sequence a protein. I know that because I asked the question one time, can't we just sequence the proteins, figure out the sequences of proteins in a cell? They all laughed. They said, oh no, that's going to be too hard. We would need all this mass spec stuff. And just like, oh, I shouldn't have asked that question. Now I feel dumb. Of course, they were all PhD students in my class, postdocs, guys that already had their PhDs. So they, I could afford to take it from them, because they're a smart crowd. So practically speaking, let me just say that, practically speaking, you can't. I mean, if you tried real hard with modern day machinery, the way it'd probably be done today is they would get the sequence, they would put it in DNA, and then stick the DNA in a bacteria, and that's the way they'd make it. So yeah, even the way we would do it today would involve DNA.
That's what James Tour says, is that all they do is buy DNA from someplace else. They don't create it. They just inject it.
Yeah, it's very hard. And see this, now that you're learning this, there was some flaw in what R.N. Ra was telling us. He said there were all these peptides in the first life. I'm just like, well, that'd be no good if you don't have DNA that codes it. See, I just didn't feel like throwing that in there because, one, I didn't want to clog up the emails. And the other thing is, the most important thing he says, there are no effing common ancestors for proteins. And I said,
well, and even if there were, proteins by themselves are not sufficient if they have to have the DNA that goes with it. Is that?
Yes. Okay, there's an example of this. We have what we call enucleated cells. Enucleated. Enucleated means they took the cell and they took out the DNA. Now, the cell can live for a while without that. In fact, there are experiments where there's just the mRNA and the protein, and the cell was happily able to continue, but it couldn't replicate. It didn't immediately die. And in fact, sometimes it would actually undergo mitosis, which is surprising. They could make more cells. They did this a lot in developmental biology. But then it would eventually stop because it would run out of mRNA, and it couldn't make any more proteins. So I'm just telling you that as a technicality, it'll still survive for a while. It can survive in isolation for a little bit, but at some point, if it needs to manufacture new proteins, like 'cause sometimes the proteins wear out, if you need to make a new one, just kind of, like your tire wears out, if you need a new one and you lost the recipe, and once it's gone, you're out of luck. And so there's that song, MacArthur's Park, and I'll never get that recipe again.
Oh, no. Yes, I know the one.
Yeah, all right. Oh, boy, we're making ourselves feel old. That was from the old disco era of the 1970s. MacArthur's Park, Donna.
What you got going on tonight, Sal? You said you had some lessons prepared for me.
Yeah, well, this is part of it.
Good.
But you see, the way you can remember things is why is DNA important? If you don't have it, you never have that recipe again. And actually, I do emphasize humor as remembering things. So that's why I sometimes these rabbit trails are helpful for nice memory devices. Donna Summer, that's her name. Okay, I'll never have.
No, she did not do that. It was way before her. Let me Google it if you really want to know.
Well, that was the guy who played King Arthur, Richard Harris. It was Richard Harris who did Camelot on Broadway. MacArthur's Park.
Right, done by Richard Harris.
Yeah.
Okay.
And Janet Duttridge is here. I've kept busy trying to get all your streams to thumbs
You're even
up.
right about Donna Summer. Why was I remembering somebody like Three Dog Night or some old? Anyway, sorry. Hi, Janet.
Here we go. Can you say a quick prayer for Janet?
Sure. Heavenly Father, thank you for sisters in Christ. Lord, you say that when we have fellowship in your sufferings, that we will also have fellowship in your resurrection. And I know Janet and myself and even Sal have had plenty of sufferings. And sometimes it's because we stand up for what is right, which means fellowshipping in your sufferings. So, Lord, we just look forward to fellowshipping in your resurrection. We cannot wait for that day. Thank you that even though we're not worthy, that by your blood we can enter in. Help us accomplish the good works on this planet that you have established for us from the foundation of the earth, Lord. Help us endure till the end, that we could hear you say, good and faithful servant. In Jesus' name, amen.
Amen. And Janet Duttridge says, I love that song, but not by Donna Summer. I'm blanking the original. It was Richard Harris. Richard Harris.
It does say Richard Harris. I just don't remember it like that.
Oh, she said, I know Glenn Campbell did the song with a large famous orchestra that was beautiful. And Janet just said, thank you, Cindy.
You're welcome, Janet.
Yeah, Richard Harris played King Arthur in the Broadway version of Camelot.
You're right. It has a relationship. It's from the movie Camelot, which I never knew. I just read it.
Okay, so why don't I show you all the steps in, there is a diagram that, okay, so I showed this here, which is the NIH, but it's going to be easier to remember this if I start showing you some videos. I got cleared copyrights of certain stuff, and I'll play them, and we'll just keep watching it until you get it. And that's how I learned it. I sometimes watch the same video 20 times, and even after the 20th time, I'm still learning something. Okay, so
it's kind of like reading the Bible. Each time you read it, you catch a nuance you missed the first time.
Exactly. So, I mean, this is actually, this is a great way. In fact, when you study biochemistry, you see just a lot of repetition. The professor said to just use a index card. And if you're riding the bus or whatever, you can read it. Yeah, Janet says there's an echo here when Sal talks. Unfortunately, the cure for that is having those earbuds. Otherwise, Cindy can't hear me if my volume's not enough, and that does cause feedback. But when I play some videos, we can suppress that. And she also does mute herself. So, okay. So we're going to play I'm going to play a video a few times. We'll learn some terms. So we'll start with DNA transcription.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It
This is
begins
a little
with
soft.
a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
I can play it again, but your thoughts?
Stunning! In real time. It's reading something that fast. And creating a copy with building blocks just streaming in, like coincidentally from a soup? No. They're special building blocks.
You could see how difficult the origin of life problem is. Okay, that's just transcription. Why don't I play it again and just
Those
try to
yellow
care?
building blocks, where are
The
they
yellow,
coming from?
they have to be they have to be synthesized and then transported to uh, the vicinity.
And placed in exactly the right spot.
Correct. Okay, so there's machinery that synthesizes the raw parts. So when you have Scrabble letters, our Scrabble letters are the alphabet of 26 letters in the English. In RNA, what's the RNA alphabet?
ACTG.
No, the DNA alphabet is ACTG.
That's what I
What's
just
the
said.
RNA? Okay,
Oh, ACUG.
ACUG. Very good. Very good. Bravo. All right, so you have the ACUG RNA uh, ribonucleotides floating around, and there's a machine. Okay, so you have this pool of RNA nucleotides. You have the machine called the ribosome, which is going to be grabbing it. Okay, so it has to wait till one is close in proximity. It grabs it, and then it can put it, and then it can continue making the string. So it has to learn how to pause when it doesn't have. It has to wait till it gets the RNA it needs.
It has to
The ribonucleotide.
detect if it's A, C, U, or G, and it has
Yes.
to put it on the rung and quickly get the next one in the speed that we just saw.
It's able to do it. It doesn't have to, but it does it so quickly. It's amazing.
Isn't it like every 3 seconds it's doing it?
I don't know what the actual speed is, but it's fast. It's a lot faster than we can synthesize RNAs in the lab. The way we do it in the lab is so brutally difficult. That's why I just laugh when Thumbduck Farina says it's easy to make any biomolecule. That's a joke. Okay, so we'll play it again.
Even if they were right there, okay, even if they didn't have to be synthesized, even if they were just right there, how can you just detect which of the 4 it is and put it in the right place in that kind of time? And just having to synthesize them and get them to the right spot. It's like, what is their explanation for that?
You need more machines. The way it's done right now in our current biology is there are all these machines that do that. So just think how difficult it is to make a machine that can do that and do it so well.
But what is their explanation for what we just saw?
Of how it originated?
Mm-hmm.
They don't have one. That's exactly what James Tour was saying.
So, I mean, how is not just where did it originate, but how is it even possible without an intelligence, without a God?
Well,
it's a faith statement.
That's why I wanted, you see, just in 2 minutes, all I had to do is show you a video. If you had minimal amount of familiarity, you'll see the problem. I would rather, this is why I say, this is just textbook. I didn't quote a creationist source.
And the yellow strand of RNA, well, and the DNA, they're just recipes. They are what turn it into flesh and blood. They're just information. They're not even the flesh and blood yet.
They are the information bearing molecules.
That's what stunned me when I heard James Tour liken it to a storage disk. The storage disk tells the computer what to do, or in a case of a factory, it would tell the factory what to do, and then you would come out with a product. I mean, it's stunning. It's basically taking information and making it into flesh
Yes. Okay, so you've declared the works of the Lord here. See, modern science has made it easy for us creationists. You see how easy this was?
3 seconds and it's like, wow, there's God.
Okay, so remember I said the anti-evolutionism handbook? This is kind of the anti-evolutionism or abiogenesism handbook. You don't have to do a lot of heavy lifting. If you're just familiar with the resources, see, this is why it's great. Where did we start? I said the central dogma of molecular biology. And that may have seemed really dry, and why am I teaching this to you? But when you understand the steps and you have these videos like here, it's going to be very easy to teach. Because this is in the textbooks. No one's going to argue with you about the operation. And all you have to do is say, how did that happen? Do you have the answers?
And how does it fit in a cell?
You'll see that as you study cellular biology. It's all very compacted. There's hardly any room in the cell. There's hardly any wasted space. It's amazing. And it can sort it all out and not get confused.
Of eternity.
So
you can go as big as you want and as small as you want. You're mute.
Okay, I unmuted myself. So what was the name of the process we just saw? Transcription. Let's watch it again. I'm gonna learn with you here. This is good for me to watch, okay?
Mm-hmm. What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
And we've been joined by Dr. Joel Duff. Greetings. Welcome. The Lord be with you. And he says, and to think it's way more complex than that. And Dr. Joel is a professor of biology. He trains, he's an advisor to PhD biology students. So he's really smart, very accomplished. And he says, love this video. Show my class this one. Show my class this one. So I'm giving, okay, there you go. There's an endorsement here. I'm giving you standard stuff, college level biology right there. So, oh, you're muted. You have to unmute yourself.
So the mRNA is still instruction. It's still
okay.
Not a protein.
Just a little correction here. It's better to call that raw RNA, there has to be more processing, particularly in eukaryotic cells, to make it an mRNA. So just call it RNA for generic. You're correct. At some point, it becomes an mRNA, but in eukaryotes, it doesn't start off as an mRNA. It's a raw RNA.
But
And it has
the DNA goes to an RNA.
use the word transcribe.
It's still not a useful thing. It's still just an instruction code.
It is it is a molecule that contains the instruction. There's a subtle difference, and the reason I'm trying to give you that wording is people will pounce on you.
But point being, it's still not a protein. It's interesting that the instruction code has to go to another instruction code before it can make a protein.
Correct.
Isn't that weird? Like, that's a really
complex
process.
Yes. Yes. And so those are good questions to ask. Why would God do that? Why would the intelligent? We may find out there's some genius reasons, particularly in eukaryotes. These RNAs sometimes are just transcribed. They never become proteins. They end up being part of molecular machines. They're really good materials for molecular machines and molecular computation. So what's the right way to say it? You said DNA copies to RNA. The right word is DNA.
Transcribes to RNA.
Good. DNA copies to RNA. It's DNA transcribes to RNA. Now I'll show you the next step. And there are many ways to state the central dogma. The most rigorous, comprehensive is a little bit gnarly. It's easier to draw it up. But if you can say DNA transcribes to RNA, RNA transcribes to proteins, you're pretty close.
Really? So you don't say RNA makes the proteins.
You say RNA translates to proteins.
Okay. Translates or transcribes?
Translates. So going from DNA to RNA is transcription. Going from RNA to protein is translation.
Okay.
I guess they figured this out to avoid confusion, which is great. Right. Again, these are just conventions, all right? So now on an exam, if someone's really anal, they'll make you use their version, but there's so many versions, it's almost kind of cruel. So let's see Professor Duff here. Professor Joel says, processing allows much greater control, allowing for greater complexity. If RNA was just translated directly, it would be harder to control. That is one reason bacteria can't do as much.
I don't understand what he's trying to say. What processing?
Okay,
and what RNA? I think he means if DNA was translated directly into a protein without going to RNA first.
I think, okay, the short of it is there are mechanical reasons this is a good way to do it. If one wanted
I'm sure there is. It's obviously the creation of a genius. I wouldn't be arguing with the Lord. I'm just saying it's double stunning. Mutant. So, there he is.
So I apologize for the softness of the video. I didn't do any audio balancing. I was so focused on clearing the copyrights on the video so YouTube doesn't give me a strike. But now we will do translation. So the first video was?
Transcription.
Okay. This one's going to be translation.
Okay.
Okay.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay, you can unmute yourself. Okay, what was the name of that process that you saw?
Translation.
Okay, so can you restate the central dogma of molecular biology now?
Well, I'd rather talk about what we just saw.
Okay, go ahead.
That I'd have to watch about 50 more times. So it talked about mRNA, tRNA,
Okay.
green things, those red things. So
Good.
if I'm not mistaken, the green things are something that transport the red things, and the red things get stuck together into the final product, which is the protein.
Okay, so let's cover this. The red is, technically we call it a polypeptide, but it can be, protein's good enough. If you want to be anal, just call it a polypeptide. Poly meaning many, and you have many peptide bonds between the amino acids. So what is a polypeptide made of? Amino acids connected with peptide bonds.
Amino acids, what?
Connected by peptide bonds. There's a certain kind of way that it's connected chemically, and they just use the word peptide bond, maybe to make it easier to see. Let me just...
I know that's in the collagen that I eat for breakfast. It says they're polypeptides.
Okay, let's see if I could find it. Peptide. See if I have a picture here. Some pictures are better than others. Okay. I like this picture here. So I'm going to show you this picture. I'm going to switch to a new picture.
So somehow that yellow RNA strand tells those green things to go get a certain red thing and connect it to the final product. Is that right?
I think so. We'll review it. Okay, I just wanted to show you what a peptide bond is. So this is the polypeptide or slash protein. I like the word polypeptide. It's a little clearer. Okay, so polypeptide. And what are all the little circles? Amino acids. This is conceptual. And so you have like here the pictures of the amino acids.
There's 21 amino acids.
20 canonical. Just think 20 canonical. There are actually more than 21 in the biosphere, but we don't have to go there. Just think 20 canonical.
That's going to be hard for me because, like I said, the protein powder that I used to take, it bragged about having all 21 amino acids, and so it's stuck in my head.
Oh, I got you. Okay. Well, you're close. So do you see the picture here? The peptide bond connects the two amino acids. Do you see it right here?
Basically, it's just a black line.
It's a black line conceptually. I'm trying to spare you all the chemical details because
But you've got two H's next to an O. So is that what connects?
Okay, so let me just show you. If you want the chemical details, this is what it actually looks like. So it's nicer if you just have the black line.
Basically, the amino acids have elements next to each other that attract each other so that they will bond.
Paleologos or Dr. Joel, if you could kill the bot. There, we just got attacked.
Hi, Peter.
Thank you, Paleologos. Thank you. You're the man. Okay, so in biochemistry class, you'd have to describe the starting state and then the end state, the process, and then you would draw this diagram up. But for our purposes...
Wait, let me look at that.
Yeah. So O is oxygen, double bonded with carbon, and then N is nitrogen.
Where did
The R...
Okay,
Okay,
what is R?
The R is the remainder group that distinguishes what amino acids it is. So your 21, rather than putting a specific set of atoms there, they just put R, which means whatever.
Okay, because I'm like, wait a minute.
Yeah,
there's no chemical named R.
Not that I know of. Maybe there is. I've never used it.
Okay.
So they call that an R group or residue. I think that means residue, R for residue. Okay. So why don't I show you the various R groups just for completeness since you brought it up. No, this is good. Amino acids. And we'll see what the R groups look like. I like Leniger because that just happens to be the book I learned chemistry from, although there are others who swear by Vought and they don't. Oh, here it is. Here's my Leniger. Okay, so each of the blue things are the R groups. So that's how it's distinguished, what amino acid is which. So those are the R groups. 1, 2, 3, 4, 5, 6.
So the blue is different, but the black is always the same because that's the initial nucleotide.
No.
Is that right?
No. The blue is different, but this group here is generally the same. You have an amino. I think this is the amino group. Let me make this bigger.
Well, that's what I said. The black are all the same, and the blue varies.
Okay, so this is the carboxy or carboxyl group. So now you're learning organic chemistry here, and I'm going to botch this up. So we have there's certain spellings of atoms when they group together, we can call them functional groups. So this is the carboxyl group. This is the amino group, I think. Carboxyl and amino. And when you connect, what happens is you connect the carboxyl with the amino group of the other, and that's what creates a peptide bond. So here it is. This is the carboxyl group here. COOH. Sometimes the H is left off. Depends on the convention.
So the H enters in from someplace else and creates the bond.
The bond has to be fused together by that machine called the ribosome. This is not a natural process. This is what James Tour, when he was saying this doesn't spontaneously happen in water. That's the water paradox. Because it
has to go through a machine.
Because it's energetically, you have to add energy, and then you can bond them together in water.
So no elements are added, just energy. Energy.
So what happens is when this bond forms, so let's go back here. Let's go back here. Oh, there you go. Ray Giordano is our resident chemist. Sir, he said, amine. I shouldn't say amino, it's amine. Thank you. So I knew a chemist would show up. See, we got a smart crowd here. We have Dr. Joel, we have Paleologos, we have Ray Giordano. So you've got people here to support you, Cindi. They're delighted to see you learning.
Make sure that Sal teaches me the right thing.
That's right.
They must also not be here just to watch me learn. They must be here because they want to be in the battle.
Well, I'd like to think they want to be here to hang out with both of us. So,
like, contend for the faith?
No, this is a nerd session. Maybe they just like nerding out on a Saturday night. Okay, I'm making you an honorary nerd. Okay, you're talking biochemistry and molecular biology on a Saturday night. You're an honorary nerd.
They want to look into the glories of the cell.
All right, so we have here, so you have an amine and then a carboxy group. They come together. The red becomes water, and then when the two join together, it becomes a peptide bond. So you'll just hear that. So when they say,
"Oh, the red became water," because you got H2O.
What?
That's what I thought, that the red became water because you got two H's and an O.
Correct. So this is what they call, okay, when water is created, it is a condensation reaction. I don't know how is that right, Ray? That's a condensation reaction. And Dr.
So
Joel
H
says,
is taken off of the top of the C.
The OH is taken off the C.
No, the H, you see the H on top of the C, the middle C?
This one?
Yes, because it's replaced by an R down below.
I think there's a mistake in the diagram. This should be an R. This is a bad diagram. I think this should be an R.
Well, that's the raw
R group.
And then down below is the product.
Oh, see, this is
doing on a Saturday night.
And Ray says, it's interesting that only 20 amino acids are used, depending on how loosely you defined it. There's many thousands of amino acids. Yes.
Really?
Yes.
Really? And only 20 of them are used?
In the process of translation, there are only 20.
Okay. So there's other things that happen besides.
I mean, kind of like, okay, canonical. I like the word canonical because that means 20 unless you have exceptions. All right. So just say 20 canonical and people will know what you mean.
Well, that reminds me of scripture. There's canon and then there's uncanonized books
Right.
that aren't accepted into the canon. There must be a connection.
Yeah. Very religious here. Canonical, 20 canonical. If you say 20 canonical amino acids, you'll be good. Okay.
Yeah, but
People,
I'm working on that.
20 canonical. All right. So I think this diagram is wrong. Shame on them. This is a better diagram then. Because
Canon, according to or ordered by law, including in the list of sacred books officially accepted as genuine.
and DNA is the language of God. So there it is. Sacred book. This
that
is a sacred
really something else.
Okay, this is a better diagram because it has the R groups. So thank you for pointing out a mistake in the prior diagram that I grabbed off the internet. So that must have been done by a graduate student who was overly tired and he made a mistake in his chemistry. So the R groups, remember the R groups are like, these are the R groups here in blue.
And
they distinguish the amino acids. R for residue. That's one way to remember it. They use the word residue sometimes.
basically they're a certain molecule.
Right. And yeah. So you have the R group. And then so this carboxyl group joins with the amine group. And so the red molecules, this molecule comes out. Where's the other? Okay, no, here it is. That's not quite right.
Drop off and become a water.
Yeah, they should have. Okay, this is another mistake in the diagram. They need to make this red, and this red, and this one blue. This is a mistake. They need to make these two red and make this one black. Because the OH joins with, oh no, I'm sorry, it's the blue. I'm sorry, they reversed the colors. The blue becomes the water. So you have the OH, they call this a hydroxyl group. The hydroxyl group combines with this C double bonded with an O. So this whole thing is a carboxyl group. So the carboxyl group contains a hydroxyl group. So the hydroxyl group connects to the H. So H, two H's and one O, you get H2O.
So the OH on the left makes that a stable molecule, and just the plain H on right there where you're pointing makes that stable. But when you put them together, those drop off and it stabilizes by hooking together.
Well, I don't use the word stable. This will
what do you call it?
I don't know. They are functional groups.
Yeah, functional by themselves.
Yeah, functional groups. And
so the machine you refer to that combines them must be a machine that pulls water out.
Correct. Condensation reaction. Condensation. Now, this is actually, it's better represented to have two arrows because it can go the reverse. Condensation makes water. Hydrolysis adds water. So if we reverse the process and put water here, you'd split it apart. Hydrolysis. So James Tour was using the word, you have all these spontaneous hydrolysis reactions. So if you have amino acids floating out there in water, they're going to split apart eventually. In addition to racemizing, all this natural degradation.
Oh, wait a minute.
He was hammering Farina over the head. He said, "You got a problem there, buddy. How do you polymerize this? Because it doesn't have an inherent tendency to stay together. It had, if you throw a protein, technically a polypeptide, in water, it has a natural tendency to break apart. The other thing is in isolation, if you have all these amino acids floating in water, let me just show you.
It's going to revert back to
Well,
the left.
Well, if you have the amino acids floating around in water, the natural tendency is not to come back together and make more water. We just know that from the chemistry.
Well, that's what I'm saying. That's where you said you would put the arrow going both directions.
Right. Correct. So what James Tour is saying, in water, if you start off with this state in water, it's going to stay that way. It's not going to spontaneously, what we call, form these peptide bonds. And the word he might use is polymerize. So by the way, so if you have an isolated unit, the amino acid, we can call that a monomer. And
that's why it's polymerizing on the right. Because it's no longer two monomers. It's now a polymer.
Right. And now there's some anal terminology. When you have 2, we call that a dimer. When you have 3, it's a trimer. Anything less than, I think, 10 or 50, it's called an oligomer. And beyond a certain number, they just arbitrarily call that a polymer. I'm sorry. People will they'll let it slide. I just want you to know there's some people that are be real word lawyers and say, no, that's not a polymer. It's an oligomer. It's
really strange how it's like the camel and the gnat.
Yeah. Yeah. So the chemists came up with these words. I don't know that it was anything nefarious, but in Origin of Life debates, some guys will get after you. I'm just pointing that out. Even I don't actually know, when an oligomer is a polymer or vice versa. But you do hear the word dimer for 2, trimer for 3, tetramer for 4, pentamer for 5. After that, they're just like, well, whatever, octamer for 8. And even I've talked to some professional biochemists who are like, what's a hectamer? And we're like, hey, Joe, how many is in a hectamer? I have to look at it. He said, I don't know what that number is. What's the hectamer?
What is a hectamer? It sounds like
it's 100. I think a hectamer is 100. And Paleologo says, no, definitely nefarious, just to confuse students.
A metric unit of length equal to 100 meters, a hectometer.
Okay, so a hectamer is 100.
That's weird. Why isn't it a decimer? That would be 10. You're right.
So you have octamers, 8, decimers, 10. I've never heard of the one for 9 or 7. Maybe because they're so rare in nature. Honestly, they are rare. So by the way, it's not limited to amino acids being the monomer units. In fact, what can happen, and later on you'll see, like even the polypeptides themselves can be joined to other polypeptides, and they'll create protein complexes that they'll call, like you heard me use the word homodimeric topoisomerase. And then the homohexameric helicase, or the homotetrameric potassium ion channel, or the heterotetrameric gyrase, or the heterotetrameric hemoglobin. But you'll learn all of this. It'll start to be natural. So once you learn this, you're going to have an easier time also interacting with people and reading literature. It won't be so intimidating.
Just naming chemical compounds.
Yes, this is like learning a new language. That's all. It's not like differential calculus where you have to get things so exactly right. Otherwise, you won't be able to execute. But here, it's just like memorizing names and understanding the naming conventions. Oh, and you're being put to sleep. That's wonderful. As we declare God's work.
I do remember chemistry. I liked chemistry.
Okay, so Peter says definitely nefarious, just to confuse students. All right. And Ray Giordano is a resident chemist. He's a professional chemist. He said the idea of life forming in the ocean, aka the prebiotic soup, has many problems, including the hydrolyzation of the peptide bonds, reversing the reaction. Exactly. You need to have confusing jargon in order to be considered a professional. There you go. All right. So that's just the way it is. And I'm just trying to help you navigate this. And when you start using these words, you'll also be a little bit more respected because you've shown to the world you've invested time to study this. So there is a benefit to your witness that you've shown the willingness to suffer through this, and it'll say something to your students. So it's more than just being able to formulate clever arguments. It's also a witness. It's like, hey, if I could do it, you could do it too. And, it'll be.
I think they'll still always find a way to mock.
Well, but for those who are seekers, there's a point you will shake the dust off your sandals. Go to those who will be delighted to hear you.
Yeah.
The only reason you listen about the mockers is to understand their arguments. Sometimes they'll help you improve your arguments.
Yeah. Or help it. My goal is to understand their arguments so I can debunk them.
And sometimes they'll give you gems, like there's no effing common ancestor for proteins. So now you understand. Okay, so now you understand the proteins, kind of the synthesis. We call this also translation. We also call it protein synthesis. Protein synthesis.
The creation of proteins.
Correct. Okay, so now let's go back here to the video.
The three proteins that are essential for life.
All right, so this is made of amino acids and it makes the polypeptides. And I had to mute you a little bit, Cindy. I'll unmute you. Are you all right?
Yeah. I'm yawning and I didn't want you to see me.
Oh, no,
So
it's okay.
The polypeptides are the red things.
Correct. This other complex here is called the ribosome. Ribosome.
And the ribosome is the thing.
Want me to show you the parts of a ribosome?
That executes the code. The ribosome is what executes the code and turns it into flesh and blood.
Is our mRNA. It has the recipe. The ribosome is going to take the recipe, and it's like the cook. So just think of the ribosome. That's
what I said. It's going to execute it.
Right. So now you're asking about the other parts. Okay, you asked about tRNA, and now this makes sense why there are RNAs, why you have that third step. You don't go straight from DNA to RNA. Let me see if we have it here.
Well, and James Tour was talking about ribose, that they buy ribose.
Okay, this is what's unfortunate. We have a lot of similar sounding names, and they're totally different. Ribose is a sugar. A ribosome has nothing to do with sugar.
Okay, thank you.
I mean, it has sugar molecules in it, but I don't know how they came up with the names. All right. So you're a chemist.
Just is a machine.
Right.
A ribosome is the cook. Takes the ingredients, puts them together, and makes the cookies.
That's right.
Mm-hmm.
Analogies are great. So now I'm going back to this. Let's go back to this here. You asked about the tRNA. So it turns out you need RNAs to make the machines. This now makes sense. Sometimes in the transcription process, you're going to make RNAs that don't become proteins. So let's look at this.
Wait a minute. You need RNAs to make the machines, but you need the machines to make the RNAs. Isn't that what you just said?
Yeah.
Which is another indication of...
Okay. So
what I would call irreducible complexity, and you would slap my hand.
Design,
Huh?
creation, a miracle.
It all had to be there at once.
Yeah, there's good argument for that.
Didn't you just say that the RNA is required to make that blue thing, and that blue thing is required to make the RNA? No, the RNA.
No, the blue thing. No, the blue thing is important at some point. But let's go back. Let me go back to the diagram for the central dogma of molecular biology. So there's a step here that's not really shown. You have the DNA. It makes the RNA that gets translated to a protein. But there's some DNAs that just become RNAs that never get translated to a protein. Some of these DNAs will create the tRNAs, these transfer RNAs.
The green things.
The green things. So the DNA.
You read in groups.
Okay, so this is a great question. Great question. Okay, so let's back up. See if I could back this up. Okay, so the yellow.
They would call junk that we are.
No, it's non-coding DNA. Not all non-coding DNA is called junk by the evolutionists. And that's such a lousy word. That's something coined by evolutionists, junk DNA. And see, they don't.
It's intentionally insulting.
Of course, they don't want to call it designed or functional.
Just like that guy the other day, he was defending abortion, saying the child was a parasite.
Yes. This is how they insult God's work. They have to use derogatory metaphors for something that is a miracle of God.
Yeah.
That's just the way, that's the modus operandi.
The MO.
That's right, the MO.
Okay, so you were saying.
All right, so you see the green is RNA, and it's still RNA, but they call it a tRNA. It's a certain kind of RNA, a tRNA.
T stands for translator, right?
Transfer.
It's the thing that takes those yellow things out of the environment and puts them onto the rungs.
No.
Oh.
Okay, you'll get it sorted out. Okay, this is all you have to do is we just repeat it.
No, we're not making rungs anymore. We already have the rungs. Now we're making proteins. Okay, so it takes the yellow things and puts them into the polypeptides.
It takes no.
It's not taking yellow things. It's taking the red things.
It's taking the amino acid. Okay, so in transcription, you're seeing those little yellow things floating around. Those were ribonucleotides.
Right.
The ACUGs floating around. And then they form this RNA,
which is the yellow strand.
Right.
And now the yellow strand
is RNA. The greens are also RNAs. So the DNA has to have this kind of RNA that is yellow, and then the other kind of RNA that is green. So the green RNAs are tRNAs. The yellows are your mRNAs. And by the way, there are so many different kinds of RNAs. I can't even keep track of them. There's the miRNA, the siRNA, snow RNAs, rRNAs.
And they don't all come from
They are transcribed from the DNA. So there's lots of RNAs that never get translated. There are lots of DNAs that will make RNAs that never get translated. And I need to tell you one.
They are all part of the dogma. They all are part of the transcription process.
They're part of the transcription process, but they never become a protein.
Because it looked like from the video that the transcription process only created one yellow strand. It didn't create any of those green things.
Correct. And there's something else I need to tell you that's really cool.
mRNA,
Hang
that
on.
it can be
Oops.
registered,
Stop.
known as a
The blue is mostly made of RNAs also. The green is made of RNAs, the blue is made of RNAs and some proteins, and the yellow is made of RNAs.
The yellow is the only thing from the transcription process, though, right? And so
No,
where
all
did
of
the blue and the green come from?
the blue, the green, and the yellow are all from the transcription process.
But that video showed only the yellow snaking out. It didn't show
Correct.
anything blue or green being created.
Correct. So I'm giving you some subtleties. This is one of the problems when we present it simply. It can be a little misleading because we're trying not to make it too complicated, but I'm giving you the picture here. The green are RNAs, tRNAs. The blue has a lot is proteins mixed with rRNAs, two R's, R, and A. And the yellow is mRNA. So let me show you the construction of a ribosome. Ribosome parts. Eukaryote is complex. The eukaryote is even just complex. So
You're breaking up a little bit. I'm going to mute or maybe stop my cam. So you're about to blow my mind, aren't you? I've been able to understand thus far.
Cindi, we're losing you. Cindi, we're losing you. I'm sorry.
I can hear you fine, and I can see the video fine.
Okay, we recovered. All right, so you see that blue thing?
Yeah.
This is an approximation of the blue thing. When you see like the 40S, that's an RNA. The 60S is an RNA. I'm pretty sure. Let me double check. I could have that wrong. Someone's going to ding me. 40S. It could be a protein. Oh, let's see. 40S.
No, you're right. It says ribo.
Okay. This 40S is made of proteins and RNAs. And I think the 60S is also made of proteins and RNAs. So let's look at 60S. It contains a 28S rRNA that is homologous to the prokaryotic 23S ribosomal RNA. In addition, it contains 5.8S rRNA and corresponds to the 5'. Okay. Just suffice to say, this is really complicated. The ribosome is made of RNAs and proteins.
Okay, so this brings up the whole issue of random. Okay, we would say that because evolution is random, and this is obviously a completely not random process, but Dr. Chris was saying how stupid we are because evolution is not random. It's controlled by selection. That's ridiculous.
Well, if he's still saying that even after I showed him that Darwin's theory of unnatural selection doesn't work. There are numerous reasons for that, and they can keep ignoring it. They're going to have to, otherwise they're going to have to start from square one. They can no longer argue that. One of the first problems is how can you evolve something that's dead? You can't select for what doesn't exist. You can't select for what doesn't exist.
And who's doing the selection? And like you said, if it's dead, there's no selection going on.
Exactly. you can't start from first principles of chemistry and physics and say there's going to be selection pressure to make these molecules. They assume that it exists. That's an assumption. I said, then I'll just say, what's the probability you're going to have a selection pressure?
Selection pressure only occurs inside living things.
Selection pressure only occurs in the imagination of evolutionary biologists.
Well, I mean, they would say, they just basically, whatever you call it, they'll take the selection process that makes the Arctic hare turn white because it's in the snow. And they will extrapolate that to include, some selection process that they turn into a foot.
So basically, this complexity arises by unnatural selection. So I said selection exists only in the imagination of evolutionary biologists. What I really meant, that's shorthand. Unnatural selection exists only in the minds of evolutionary biologists, because these steps would be highly unnatural. And there are numerous ways to prove it. Numerous experimental ways. And that's the other thing they don't have.
The fact that it happens so fast indicates there's no selection going on. It's pre-programmed. It's executing commands. It's not looking at the environment to see what would fit best.
Yeah. Okay, so I found this this is also educational for me. I found this, ribosomes are usually made up of 3 or 4 rRNA molecules and anywhere from about 40 to 80 different ribosomal proteins. Each ribosome is composed of 2 subunits, a larger one and a smaller one, each of which has a characteristic shape. And that's from Encyclopedia Britannica. Okay, so you need at least what? 44 parts to make this machine?
Yeah, I mean, every time, like the video was simplifying it to say there's 2 parts,
Codon.
A larger and a smaller.
Each codon on
But every single thing we look at apart, when you look at it closer, it has subparts. And I bet when you look at them closer, they have subparts. It just is infinite. It keeps going. Smaller and smaller and smaller.
Well, it will come down to the subatomic particles at some point. But
right.
Okay, so the blue is the ribosome. It's made of 4 rRNA molecules and from 40 to 80 different proteins. Each protein has about, on average, I'd guess around 400 amino acids in the right sequence.
And where do these come from? Okay, that's how this conversation started, because they weren't pictured. All that was pictured was the yellow snake. But you said that these blue and green things do also come from DNA. So do they come from a separate transcription process, or are they knockoffs from the yellow snake?
You have to transcribe all these things to have all the parts, all the RNA parts. Some of the RNAs don't get translated, but for the 80 proteins, you have to translate. So you need a minimum of 40 genes that get transcribed into those yellow snakes. Those yellow snakes then get translated to proteins. Then you can make another ribosome. You can't.
Okay, so you're saying those proteins make up that blue thing.
Right.
That's what you were saying before. Proteins are needed for making
proteins are needed to make other proteins. You need proteins to make DNA. You need DNA to make proteins.
Thank you.
And the chicken and the egg is even more complex, because you need proteins to make the proteins that make the ribosome. And it just said from from Britannica, there are at least 40 of those ribosomal proteins.
It's the chicken and the egg.
Oh, yes. So this is, James Tour is not even getting to this level, but people know. George Church looked at this. He said, "Yeah, if I were going to argue for intelligent design, I said focus on the ribosome." I'm like, yeah, and he's one of the most eminent scientists on the planet. So,
is he an ID proponent?
He's on the edge. Let's say he avoids the question, but he recognizes we're making compelling arguments. So let's see. What did he say? Darwin's George Church. Okay, so famed Harvard geneticist. He said,
Can you share screen?
Of one of the Discovery Institute's books. What?
Can you share your screen?
Yeah. George Church. And he said, Darwin's doubt represents an opportunity for bridge building rather than dismissive polarizations, bridges across cultural divides in great need of professional, respectful dialogue, and bridges to span evolutionary gaps. And that's George Church.
So basically, he's saying not to be partisan.
Yeah, but that's better than being, fighting us.
It's
one other
really
thing is
it's giving us the respect that we, hey, even if you're not on that side, listen to their argument.
Exactly. But he's also saying it professionally. You know why? I'll tell you why. He is redesigning the ribosome for certain medical applications to be able to make drugs. So a lot of drugs like insulin is a polypeptide. It's a protein. It's desirable to make certain so that's a medical, that's medicine right there. By making insulin, you're making medicine, because like people who are diabetic, they need to have artificial insulin.
Yeah,
so
yeah.
So we need to find ways to make artificial chemicals. Many of those chemicals are proteins. Not all of them, but many of these pharmaceuticals are proteins. And so he's redesigning the ribosome to make safer ways to experiment, because you don't want to make a bad protein and then it just spills out. You don't, because what happens is you put these new DNAs into bacteria, and what happens if the bacteria escape the lab? It could be really bad. So to add safety, he redesigned the ribosome so that it'll limit that possibility. In the process of re-engineering the ribosome, you learn a lot of how complicated this is.
I bet.
Jordana says, here, Ray Jordana says, I'm still alive thanks to insulin. God bless you. So
Now,
my mom,
Go ahead. I was just going to say, since all of this is microscopic, I mean, I don't think they can even see DNA under a microscope, right?
I'll show you.
How the heck do they do this?
I'll show you a picture, some of the best pictures we have. So
I mean, what does he work with to re-engineer the ribosome? Does he?
It's hard.
Here's
some electron microscope graphs of DNA. You definitely don't see the individual atoms.
Can we see an atom under an electron microscope?
Not atoms of that size. You might be lucky if you have some of these really big atoms and you have something in a crystal structure, you might be able to see it. But so all these models of the molecules, they are done by inference, because they're basically unseen realms, because they're so tiny.
That's what I'm wondering, how the heck you would do that.
That's where biochemistry and chemistry and physical chemistry come in. It's difficult. And we have a professional chemist here. So they have to, this is what I call real science. They're able to figure out the structure without seeing it.
It's amazing.
That's why I laugh at evolutionary biology. They don't have the technology or the ability to learn how to do stuff like this. They can just tell stories. They're good at that. But doing real science, like figuring out the structure of DNA, very hard. The first people to deduce the structure of DNA, they had to use X-ray. They had to use X-rays. It was Rosalind Franklin and then Watson and Crick used her work. They got the Nobel Prize. Poor Rosalind Franklin was ignored, and then she died because she was exposed to so many X-rays. So, yeah, it's hard.
Pretty quick.
What?
It's 9:30. I gotta go pretty quick.
Oh, okay. So we can continue this again. I hope.
Yeah.
Okay.
Anything quick that you wanted to cover that we didn't?
No, let's just stop here.
Okay.
And there's a small favor I could ask. If you can get some sort of headset or earbuds, the audio would be better for our viewers.
Okay. Let me see.
At some point for our next show.
Well, I have a pair, but we'll have to test them out.
Okay. So
see if they work.
I'd like to thank the viewers. We have a professional chemist. We have Dr. Joel, who's a professor of biology. And we have Pele Lugos, who's a student of biology. And then you have me. You have all these nerds hanging out with you, Cindi, tonight on a Saturday night.
Nerds rule.
Yeah, nerds rule. Okay, so let me just start. I think we could. I'm going to just play the end credits. Do you have any closing words?
No, I'm untangling my earbuds, so when we're off camera, we can test it out.
Okay. All right, we'll close the stream, guys. I'm not going to play the end credits because Cindi has to attend to something. Take care. God bless you. And thank you all for joining us today. Tomorrow, I'm going to have another show with Dr. Sandy Pigeon, a retired Navy SEAL. We're going to be shooting the breeze on military science and military history and playing armchair field marshal. Take care. God bless you all. Have a good night.
Good night.